Good afternoon. First, I would like to thank the organizers for their kind invitation. It's a great honor and pleasure to be part of this conference. Today, my talk is entitled A New Conceptualization of Human Visual Sensory Memory. Memory is an essential component of cognition. Without memory, we cannot retain information, hence we cannot learn anything new. We cannot even carry out our day-to-day tasks if we don't remember or if we cannot remember our past states or future goals. In fact, disorders of memory have huge personal and social costs. Several lines of research show that memory is not a unitary process. And the Atkinson-Shiffrin model, proposed in 1968, shown on this slide, still forms the foundation of our understanding of human memory. It is composed of three distinct stores. First, information is stored in sensory memory, whose visual component is also called iconic memory. Iconic memory has a very large capacity. However, its contents decay very fast, within few hundred milliseconds. Hence, the visual system has to transfer a subset of the contents of iconic memory to a bit more durable store, in this case, which is short-term memory. Short-term memory can retain information longer, in the order of minutes. However, its capacity is severely limited. In fact, short-term memory constitutes one of the major information processing bottlenecks in human cognition. Finally, short-term memory interacts with long-term memory to transfer some of its contents for long-term storage. Long-term memory has very large capacity. Remember all the things you've learned in your life that you can still retain, and also very long retention time. Things that you learn in your childhood, you may still retain today. The time course of long-term memory can be a lifespan. Now, since its inception of this model, there have been lots of experiments and studies on these different components. While the short-term memory and long-term memory components underwent significant changes from their original formulations based on the data, sensory memory or iconic memory remained largely unchanged. It was discovered by George Sperling in 1960, and research established four of its fundamental properties. First, as I mentioned, it has very large capacity and its contents decay very rapidly. Also, its contents are susceptible to masking, and its coordinate system, or the reference frame, is a retinotopic reference frame. So let me go one by one and explain these properties. To study experimentally memory, one can use a letter array and place randomly selected letters in an array, and also use a queue to ask an observer to report a subset of the letters in the array. Here, the queue indicates one of the rows in the array, and the task of the observer would be to report the letters in that row. In this case, it will be RTYHZL. And of course, if the array is visible out there, I don't need any memory. I can just read out from the stimulus the contents of that row. Hence, to adapt this experiment for studies of memory, what usually one does is to present that letter array briefly and turn it off. After it's turned off, it needs to be stored in memory, and the required contents need to be retrieved from the contents of the memory. And here the queue timing plays a major role in assessing how long one can retain information in memory. The fact that if I present the letter array and the queue, the arrows right after, then all you have to do is just retain information until the queue arrives. It's soon, then you don't have to retain information that long. If the queue comes with a larger delay, then you have to retain information for the duration of that delay. Hence, the queue delay is a proxy for the retention time required for memory. So to illustrate that, in this example, I will briefly flash the array. And after that, the queue will show randomly one of the rows. And the task is to report the letters in the array for that particular row. So in this case, the queue selected the row second from the bottom, and it came with a fairly long delay, at least for the time scale of sensory memory. Probably most of you couldn't remember any of the letters. Maybe you could remember one or two letters from that row. In contrast, if I present the queue with a short delay, the probability of reporting correctly the letters from that row is much higher. And this can be illustrated by the classical memory curves. Now, these data, each panel is data from one subject, and the x-axis is the queue delay, which, as I mentioned, is a proxy for the retention time for the memory. The y-axis is normalized performance, and the higher the performance, the more items you can retain from the memory array. As these curves show, when the queue delay is short or retention time is short, then the performance is very high. On the other hand, as the queue delay or retention time is increased, performance drops rapidly here as an exponential and reaches a plateau. So these typical experimental results show the first two properties of sensory memory, in that it has a very large capacity, shown very high performance levels for short delays, but it contents decay rapidly, shown by the rapid decay within few hundred milliseconds until it reaches another plateau level, and this plateau level indicates the performance of short-term memory. Another property of sensory memory is that its contents are susceptible to masking. What does that mean? If I present the letter array, and after the letter array, if I present another stimulus at the location or near the location of that letter array, what happens, the transients generated by the second stimulus will interfere with the storage into memory of the letter array. Here, for example, the second stimulus that comes, those X's in the boxes, is the mask. And when it is presented right after the letter array, if I compare that condition to the case where the mask is not presented, your performance will be much lower in the presence of the masks. In other words, masks or stimuli presented briefly after, at or near the location of memory items, interferes with the storage of the items into sensory memory. In other words, it disables sensory memory. And this will be an important property in our model as we build it. Now, the final property of sensory memory that I have mentioned is that its coordinate system, or the reference frame, is a retinotopic reference frame. So what is retinotopic reference frame? If we look at the diagram on the left, it shows how a stimulus is mapped and represented in early visual areas in the brain. We first start with the eyeballs, and the optics of the eye maps the same way a camera maps an external stimulus into its back plane as a two-dimensional image. The projections are such that the neighboring positions in space are mapped to neighboring positions in the retina, so as to create a two-dimensional image of the external world. Now, the interesting thing is that these neighborhood relationships are preserved through precise wiring from the back of the eye to the early visual cortical areas, so that the representation of the stimulus in the early visual areas also preserve these 2D image-like representations. Since these representations reflect how the stimulus is projected on the retina, they are called retinotopic representations, or the retinotopic organization. And research shows that sensory memory's coordinate system is based on a retinotopic reference frame. What does it mean? Let's look at the diagram on the right. Let's say we have an object in space whose coordinates are here, x sub s, y sub s, and z sub s. And let's say the object is stationary. Then its image will be projected at the back of my eye on the retina, and its retinotopic coordinates will be xr, yr. Now, assume that I move my eye so as to direct my gaze towards that object. Then the image of the object in retinotopic representations will shift to the central part of the retina, the fovea. So the retinotopic coordinates of the object will shift from here to here, even though the spatiotopic coordinates of the object did not change. In other words, in a retinotopic reference frame, each time there is a relative motion between the eye and the external world, coordinates will shift. What is the implication of that for vision and memory? Under normal viewing conditions, we make several eye movements. And this, for example, assume that I'm sitting in my office and those yellow arrows indicate my eye movement scanning pattern. If sensory memory is retinotopic, it means that each time my eye fixates somewhere, it registers a copy of the scene according to the retinotopic coordinates. And each time my eye moves, as I showed in that simple example, that will be shifted in retinotopic coordinates. And if sensory memory preserves the previous representation of the space, then these will be superimposed. As a result, the information coming into a sensory memory, whose reference frame is retinotopic, will be all shift and mixed up version of the external environment. And so the information will be highly messy and not very clear about the contents of the environment. This is a problem even if I do not move myself. Let's say I'm stationary, but there's a moving object in the scene. What's going to happen when the object is in a given position, say here, it will be registered in this retinotopic coordinate system at this position. And as the object moves to the next position, then its coordinates will shift in retinotopic representation and so on. So as the object moves, then you will have basically a highly blurred, shapeless representation of the object in retinotopic representations. And this is called motion blur or moving ghost problem. Now these pose a major challenge for the operation of sensory memory or perception. And these were the major challenge was already noticed back in 1980s. Haber wrote a target article in Behavioral and Brain Sciences entitled The Impending Demise of the Icon, a critique of the concept of iconic storage and visual information processing. And Haber's main points were, he acknowledged the empirical evidence for iconic memory, but given that it creates more problems than it solves, he argued that this evidence is irrelevant to perception. And the issue is, given that our eyes and heads are rarely motionless, and given that many objects in the environment are in motion, a retinotopic iconic memory would interfere with perception by blurring and by improperly integrating inputs. So it creates problems. It's not solving problems, but it's creating problems. And that leads us to the puzzle. Well, there's lots of evidence showing that there is a retinotopic sensory memory. But its implications are that it will mess up the incoming information. Then the question is, since short-term memory gets its contents from sensory memory, if the contents of sensory memory are all messed up, how does short-term memory get information? How does it transfer to long-term memory? So how do we learn anything if we cannot get clear information from the environment? So Haber couldn't offer an alternative. And also, in normal viewing, most of the time, we do not experience what a retinotopic sensory memory implies. Our perception is not highly blurred, fuzzy, but instead our perception is stable, doesn't move when I move my head or my eyes, and it's in general quite clear. So how to reconcile properties of sensory memory with normal vision? So this puzzle has been persisting for a long time, since 1980s. We've been studying this problem during the last 2 decades, and we felt that in order to provide an answer to the question, we need to actually provide answers to 3 related questions. The first one is, what purpose does a retinotopic sensory memory serve? There's a lot of evidence that it is there, but when we analyze it, we see that it doesn't solve anything, but it creates lots of problems. So why is it there? Second is, how does the visual system control detrimental effects of retinotopic sensory memory? In our answers, we will argue that actually retinotopic sensory memory does serve a purpose. There's a reason for it to be there. But then to claim that, we have to show that there are other mechanisms that control those detrimental effects, avoiding the massive blur and improper integration and superimposition of inputs. Finally, let's say we are able to control a retinotopic sensory memory, but we already know that a retinotopic sensory memory cannot operate when there is relative motion between objects in the environment and the subject. So how does the visual system process and store information non-retinotopically over space and time? Our answers to these questions are summarized here. What we propose is that sensory memory is there. And if you look at the problems associated with it, the problems arise when there's relative motion between the environment and the retinotopic representation. So, but those problems disappear if the stimulus is stationary. So we propose that retinotopic sensory memory is there to store retinotopically stationary stimuli. However, when there's relative motion, then retinotopic memory messes up its contents. So to avoid those detrimental effects, we propose that visual masking disables the operation of retinotopic sensory memory whenever there is relative motion between the scene and the retinotopic representation. And we propose that that is done by visual masking mechanisms. Finally, how does the visual system process and store information non-retinotopically over space and time? We suggest that it is using motion grouping based reference frames instead of retinotopic reference frames. This leads us to the following model, which is a modified version of the Atkinson-Shiffrin model. So the black part of this model is identical to Atkinson-Shiffrin model. External input going to a retinotopic sensory memory and going to short-term memory, interacting with long-term memory. What we are proposing is a second component in sensory memory. And the major difference between what was already in Atkinson-Shiffrin model and what we are proposing is the reference frame. This component has a retinotopic reference frame, whereas this component has a motion-based reference frame. And I will show the part of the experimental evidence that led us to the development of this motion-based reference frame for non-retinotopic sensory memory. But before I do so, I would like to highlight the importance of coordinate systems or reference frames in general in science. So in physics, it's very well known. Consider, for example, planetary motions. Now, to describe these trajectories, you need a coordinate system, and the coordinate system, a priori, can be placed anywhere you like. In the geocentric model, the coordinate system is placed on Earth. And then if you compute the motion of the planets according to that coordinate system, you obtain trajectories as shown in this figure, which are quite complex. On the other hand, if you move your coordinate system from Earth to Sun, as you can see now, the trajectories of the planets are much simpler. So normally you can pick any coordinate system and you can always transform mathematically from one to the other. But the key point here, if the choice of the coordinate system is congruent with the laws of the phenomenon you're trying to model, the resulting expressions are much simpler compared when the coordinate system is not congruent with the fundamental properties of the phenomenon you're trying to describe. We also know that reference frames or coordinate systems played a major role in the theory of relativity. Unfortunately, in cognitive sciences, reference frames or coordinate systems are not as much appreciated in physical sciences. However, I would like to highlight at least one researcher who placed coordinate systems at the center of some of his theories. That's Jean Piaget. Jean Piaget's theory of cognitive development suggests that infants are born first with egocentric reference frames and coordinate systems. What's an egocentric reference frame? It is one that is based on the subject. It could be on the head of the subject, on the body of the subject, or on the eye of the subject, as in retinotopic reference frame. These are all egocentric reference frames. What Piaget observed and proposed that through developmental stages, the brain starts to generate and use exocentric reference frames. What are exocentric reference frames? Those are the ones that are placed outside the subject, exo. So it could be a reference frame placed on some point in space or on some object. And in Piagetian theory, the switch from egocentric to exocentric reference frames plays a major role both in the sensorimotor stage and also pre-operational stage. Now coming back to our problem, sensory memory, and our question is, what is the appropriate reference frame for sensory memory? As I highlighted by examples from physics, if you pick a coordinate system which is congruent with the phenomenon that you're trying to analyze, things become much simpler, the analysis becomes much simpler. Hence, part of the job of the brain is to pick coordinate systems that will simplify the analysis of stimuli. In the case of sensory memory, we've seen that retinotopic reference frame can be okay if everything is stationary, but when things are moving, it's not a good coordinate system. So what is then the appropriate coordinate system when there's relative motion? Our suggestion is that the coordinate system will not be a static coordinate system placed somewhere in a fixed location, neither on the subject, like in retinotopic, it's not egocentric, it should be exocentric. Plus, it should not be fixed exocentrically, but rather it should follow motion patterns. For example, if I can track the motion of this stimulus, and if I place a coordinate system that moves according to that motion pattern, then that coordinate system will stabilize the moving stimulus, hence making its analysis much simpler than if I had a coordinate system that was stationary, which is not congruent with a moving object. Similarly, our proposal is that if there are multiple objects moving in the environment, it won't be a single coordinate system or reference frame because we have different motions. So we propose that the visual system parses different parts of the scene and detects motion patterns and then places moving reference frames for the different motion directions. So, now, what is the evidence for that proposal? As I said, during the last 2 decades, we conducted several studies to come up with this solution. And we used 3 main experimental paradigms, Turner-Spiegler displays, sequential metacontrast, and anorthoscopic perception. I don't have time in this talk to go all the evidence that we collected over the years. What I would like to do today is talk about the first experimental paradigm, the Turner-Spiegler displays, and show you some of the evidence that support the notion of motion grouping-based reference frames for sensory memory. Now, what's a Turner-Spiegler display? A typical Turner-Spiegler display has 2 frames, frame 1 and frame 2, separated by a blank interval for a duration, which is called interstimulus interval, or in short, ISI. The frames usually contain simple geometric shapes, and in this case, for example, we have 3 vertical bars. Once I position my stimulus in the first frame, then I build my second frame, in this case, for example, taking this and just moving it here by the same distance as this one here to obtain the second frame. So the result is that these 2 are identical for the 2 frames, and this one is displaced here. Now, what kind of percept will I generate if I present you with a Turner-Spiegler display? That depends strongly on the interstimulus interval, or ISI. Here is an example. Instead of bars, I will use 3 disks and construct my Turner-Spiegler display, and I will display it at a very short ISI. Now, if the video frame rates didn't mess up the display, what you should be perceiving there, the 2 elements that were common in the 2 frames appear stationary, static, and the element that I displaced from one edge to the other edge appears to move. So that is called element motion, and that's the percept that we get when the interstimulus interval is short. If we make the interstimulus interval long, we have a very different perception. And again, if the video rates did not mess up the stimulus, what you should be seeing now, the 3 disks moving as a group in tandem. All of the disks appear to move, so the 3 move left and right as a group. So that's why it's called group motion, which occurs when the ISI is long. So our goal is, in using the Turner-Spiegler display to study reference frames, is that we can obtain different motion-based reference frames according to the motion correspondences that we obtain in the Turner-Spiegler display. So let me show you those motion correspondences that serves as reference frames. So in the element motion, we have seen that these 2 elements, the common elements, appear stationary, and the element at the edge appears to move back and forth. Hence, the motion correspondences between the elements in the 2 frames are shown by these red arrows. This corresponds to this corresponds to this. This is the motion correspondence in the element motion case. If we keep everything the same, if we make the ISI a bit longer, as we have seen, you get a very different percept. And so, in this case, the 3 stimulus elements appear to move as a group. Hence, this element appears to move here, this element appears to move there, and so on. So we will use the Turner-Spiegler display as a tool to establish these motion-based reference frames, and then we will study if and how sensory memory uses these reference frames instead of retinotopic reference frame. So to do that, we designed the following experiment. We built a Turner-Spiegler display by using vernier elements. Vernier stimulus here, as shown, are basically two thin bars with a small gap between them. The two bars may be aligned or may be offset, shifted a little bit to the left or a little bit to the right. So in a first experiment, what we have done, we took a Turner-Spiegler display and composed of verniers, but only one of the verniers had an offset. And we call that the probe vernier. So the offset in each trial could be leftward or rightward. And what we did in the experiment, asked observers to report the perceived vernier offset for elements in the second frame. So in one session, they sat down and reported the perceived offset for element 1. In the other one, perceived offset in element 2, and in the other one, perceived offset in element 3. You may say, well, these elements in the second frame do not have an offset, so what is it that they are reporting? The key here, the contents of the first frame are stored in sensory memory and then are integrated with the second stimulus that comes within the time frame of sensory memory. Hence, if I put a stimulus here, then I will predict it that it will integrate with the contents of the memory, the contents of this memory will integrate with the second stimulus. The question is, which one? And the answer to that is depending on its reference frame. If the reference frame of sensory memory is retinotopic, then it should integrate according to its retinotopic partner. In this case, it will be this with this. They sit at exactly the same retinotopic locations. But instead, if it is based on motion-based reference frames, then the integration should be according to motion-based reference frames, not retinotopic relationships. So based on that logic, then we can derive the following predictions. So let's look at first the predictions of the retinotopic hypothesis. If the subject is reporting element 1, and again, the task of this, the offset of this trial to trial is random, left or right. And the task of the observer is to report whether they perceive this vernier shifted to the left or right. And we compute the agreement between what the observers say and what the probe vernier shift is in each trial. And we plot here percent in agreement with the probe vernier. So if the reference frame is retinotopic, then this element, which is stored at this location, should be integrated with its retinotopic partner in the second frame, namely this element. Hence, observers should perceive the offset of the probe vernier in element 1, but not in elements 2 and 3, because it shouldn't be integrating. They are sitting at different retinotopic locations. Hence, if I plot the performance of the observers in this experiment, the peak performance should be for position 1, and the other ones should be at or near chance. And the retinotopic hypothesis its prediction does not depend on ISI, because it's only based on retinotopic relationships. On the other hand, the non-retinotopic hypothesis, the prediction depends on ISI, because as we have seen, based on ISI, we have different motion correspondences, hence different motion-based reference frames. In the case of element motion, the central element, its motion correspondence in the second frame, since these appear stationary, is element 1. So non-retinotopic motion grouping-based hypothesis makes the same prediction as the retinotopic hypothesis in the case of element motion. We cannot use this condition to distinguish between the two hypotheses. However, in the case of group motion, when ISI is long, and when you see the Turner-Spiegler elements moving as a group, as we have shown, the red arrows capture the underlying motion correspondences. Now the central element appears to move to element 2 in the second frame. Hence, the prediction of motion grouping based reference frame or coordinate system for sensory memory is that this should be mainly integrated with element 2, because integration occurs along this reference frame and not along a retinotopic reference frame like this. Hence, the performance, if I look at observer's performance in the experiment, the peak should be at element 2 and not element 1. And here are the results. The x-axis is the position of attended element, the one that subjects report in the second frame. And the y-axis is percent of responses in agreement with the probe vernier offset direction. And the red curve is for element motion, short ISI, and the blue curve is for group motion. As we have mentioned, for element motion, both the retinotopic theory and the motion-based grouping hypothesis predict the same thing, that the peak would be in element 1. So it's kind of a control condition for us, and that's exactly what we observed. The condition that distinguished between the two hypotheses was the group motion case, where retinotopic hypothesis would predict a curve very similar to this, whereas non-retinotopic motion grouping-based hypothesis predicts that the integration should occur for element 2, hence the peak should occur at element 2. So that is one of the first experimental evidence to support motion grouping based reference frame in sensory memory. And we have conducted many more experiments. I will not go through the results, by placing the probe vernier in different locations, adding other verniers, so to test whether the two vernier offsets integrate or not. And in fact, all these experiments supported the same finding, in that the representation and integration of information in sensory memory occurs mainly according to motion grouping based reference frames and not retinotopic reference frames. So those experiments demonstrated that important concept for something like vernier offset discrimination, which is related to our acuity in position or computing edges of shapes and so on. One may also ask, how about other visual dimensions, for example, motion perception? And indeed, we tested the same hypothesis for motion perception and found very similar results. Here is a summary of the experiments, how we tested the reference frame for motion computation. We used 3 disks for the Turner-Spiegler display, and we used these 3 disks in the Turner-Spiegler display to generate either retinotopic or non-retinotopic reference frames. Now, to test how motion is computed, we introduced these small dots, and we made, again, distinct predictions according to retinotopic and non-retinotopic hypothesis. So let's first look at the prediction of the retinotopic hypothesis. If the motion of these small dots is computed according to a retinotopic reference frame, then the red arrows here show the retinotopic reference frame correspondences. Hence, if I represent my stimulus according to these retinotopic reference frames, this will be the first frame corresponding to these 3 disks, these locations, plus the dots inside the disks. Now, in the second frame, this one moved here, and this dot moved here, this dot moved here, as you can see, and this dot remains the same here. In the third frame, now this element moved here, this dot moved here, and this dot moved back, and the fourth frame. So if motion of that small dot is processed according to a retinotopic reference frame, as you can see, typically motion perception occurs by matches to the nearest neighbor. This will be matched to this will be matched to this, and back. So this dot will appear moving up and down. This dot will appear moving left and right. That's the prediction of the retinotopic hypothesis. And again, it's independent of ISI because it only depends on retinotopic coordinates. On the other hand, the non-retinotopic motion grouping based hypothesis its prediction depends on ISI. In the case of element motion, as we have mentioned before, these elements have the same reference frame as shown in red arrows because they don't appear to move as in the retinotopic case. So for element motion, the prediction is the same. Retinotopic or non-retinotopic hypotheses have the same prediction. We are not using this condition to distinguish between the two hypotheses, but it's kind of a base experiment to make sure that condition indeed produce the same result to support simultaneously both hypotheses. And then we go to the next one to select between the hypotheses because that's where the predictions differ. In the group motion, as you may recall, this element appears to move here, back here, back here. So the motion of this small dot should not be computed according to its retinotopic neighbor, but along this reference frame, so according to neighbors that are generated by this moving reference frame. So what would that be? The first frame, again, copy of this. Now in the second frame, as you can see, the dot positions are like this. This one moved here, but also the whole three disks moved together. So if my reference frame moves with these Turner-Spickler disks, so the reference frame for the central dot over here, which was initially here, when I move it, it will come here. So when I compute the motion of this dot, its nearest neighbor is not this or this anymore, since the coordinate moved with it. Its nearest neighbor is this one. And if I continue, again, the reference frame moves with the stimulus, the nearest neighbor is this one, and so on. So as you can see, the prediction in the case of group motion for non-retinotopic hypothesis, you should be seeing actually the small dot inside the central disk as rotating, rather than that motion being decomposed in one disk up and down and another disk left and right. That's the prediction that differs from the retinotopic hypothesis, and that will be the critical prediction. The good thing about this, the stimulus is so simple that even before collecting formal data, we can just look at it and figure out which hypothesis is correct. Here are the demos, again, hoping that the timing of displays don't mess up my demonstration. First, I will show you element motion case where both theories or both hypotheses make the same prediction, retinotopic or non-retinotopic. You should be seeing one dot going up and down and one small dot going left and right. All right, so here, this dot appears to move up and down. This dot appears to move left and right. Both theories predicted that. We observed experimentally. So far, with this experiment, both hypotheses survived. Now, the critical experiment where we will test the hypotheses against each other. Will we see the same thing, one up, down, left, right, or we will see a dot in the central disk that undergoes a rotation? If you fix your eyes and observe the stimulus, the dot inside the central disk, you should be seeing it rotate rather than having one dot going up and down, one dot going left and right. And again, the demo will work if the timing of the displays on my computer or the display over there, if the timing is correct. So that was based on the observation, and then we formally collected data. We had the group motion case and element motion, and we even had a control condition with no motion at all. Everything was static except the dot. So the prediction for these two conditions, both the retinotopic and non-retinotopic hypotheses predict that you should not be able to see rotation. You should see up and down, left and right motions. The task of the observer in this experiment was to report the rotation of the dot. So if they are not seeing a rotation, if they see up and down, left and right, they cannot report it correctly. In some trials, it will be clockwise. In some trials, it will be counterclockwise. And hence, all theories predict predict that, or all hypotheses predict that these two conditions, observers should be at chance, and here is the data, it's 50% is the chance level, so indeed they are at about chance. The prediction for group motion, retinotopic hypothesis, predicts that this one should be also at chance. Non-retinotopic grouping-based hypothesis predicts that this should be significantly above chance. That's in fact what we found. So these experiments generalize non-retinotopic reference frames from shape boundary computation to motion computation. Another area that we have tested is attention and visual search, because attention is key in transferring information from sensory memory to short-term memory. So the question we posed is the reference frame of attention a retinotopic reference frame, like it's proposed in many theories, versus non-retinotopic reference frame that we have recently discovered. A typical experimental paradigm to probe attention is the visual search paradigm. So here's an example where we have stimuli defined on two future dimensions. One is color, red or green. The other is orientation, vertical or horizontal. Here we designate one combination as the target. In this example, for example, the target may be horizontal green bar. And the rest, the other combinations, serve as distractors. So in some trials, the target is present. In other trials, the target is not present. The task of the observer is to report whether the target is present or not. In a conjunction search, like this one, where two futures, you have AND of two futures, logic Boolean AND operation, this is a very time-consuming, attention-dependent process. So most people believe that these attention maps were retinotopic. To test that hypothesis, we used the same logic with the Turner-Spiegler displays, and we placed the search patterns within the Turner-Spiegler displays and studied whether it has a retinotopic or non-retinotopic reference frame. And our results again indicated that attention and visual search also operate according to motion grouping-based non-retinotopic reference frames. So this new reference frame coordinate system that we have discovered is quite general, and it applies to various visual processes. So to summarize then, evidence from Turner-Spiegler displays and plus the other experimental paradigms that I haven't shown today indicate that future processing, storage, and integration are carried out according to motion grouping-based reference frames. And motion grouping-based reference frames, by definition, are non-retinotopic reference frames. Hence, given the empirical evidence for the third question and its answer is based on this and similar experiments, which then led us to formulate this model where, again, we have the basic Atkinson-Shiffrin model in black and the new component. Now, how does this model work? At a given point, when I inspect the scene, I may be, for example, following a target, making that target stationary through smooth pursuit on my fovea. And during that time, the rest of the scene, since my eye is moving, the stationary objects in the scene will be drifting. So in normal viewing, in general, we will have some stimuli that are stationary with respect to our retina and other stimuli moving with respect to our retina. And what happens here is the parsing of those two inputs. Our claim is that for stimuli that are stationary with respect to retinotopic coordinate system, then they go through this classical retinotopic sensory memory, and that's where they are processed. A retinotopic reference frame for a retinotopically stationary stimulus makes total sense, and hence it's processed according to a retinotopic reference frame. Therefore, all the evidence for a retinotopic sensory memory is valid. And the reason we had previously so much evidence for a retinotopic reference frame, but not non-retinotopic reference frame, because in a typical laboratory experiments on memory, stimuli were static, and therefore it went through this pathway, and repetitively it showed a retinotopic reference frame. However, for those objects that have relative motion with respect to retinotopic reference frame, as we have seen, if they were to go here, they would completely be messed up. Hence, something needs to stop them going through that pathway. And our proposal is that's through mechanisms of visual masking. And I don't have time to go through all the visual masking mechanisms and models, but remember the stimulus that I show you. That was a letter array, and then another stimulus appeared on top of the letter array, or it can also appear nearby. So whenever there are those transients, the masking model essentially prevents access to this element. So those stimuli that are not stationary with respect to retinotopic reference frame, this element is basically turned off through visual masking mechanisms. And the storage and perception of those elements in relative motion occurs through the new pathway that we have proposed as part of sensory memory, this red one here. So that pathway, the visual system will first parse different motion patterns in the visual scene and then launch reference frames that go along those motions, thereby stabilizing those moving patterns with respect to those moving reference frames, and hence having a coordinate system which is congruent with the properties of the stimulus. The stimulus is moving, and hence the coordinate system is also moving along the stimulus, thereby making it relatively stationary according to this new reference frame, thereby making its storage and processing much, much simpler. So in summary, I would like to point out to this reference paper, which summarize our findings up to 2016. We had other papers published after that, but I highlighted here the summary reference because it covers the background information and gives quite a tutorial introduction to this new modified sensory memory model. Thank you very much.